Good morning. This is John Morris, Director of the Knight Alzheimer's Disease Research Center at Washington University School of Medicine, as well as its Memory and Aging Project. And I'm providing this introduction to our Knight ADRC participants meeting, which will take place virtually on Saturday, November the 14th, 2020. So I apologize for this virtual presentation. We always look forward to seeing all of our participants and their family members when we come together, typically in June for our participants meeting and breakfast. And the interactions are something that all of us at the Knight ADRC cherish with our participants. Plus, another downside to the virtual presentation this year is there is no food and particularly no bacon. So we hope that you, if you're at home or watching this, will be able to enjoy the breakfast of your choice. Well, 2020 has been a momentous year for the Knight Alzheimer's Disease Research Center. In no small part because of the COVID-19 pandemic, but many more developments have occurred as well. And so I'm going to try to update all of our participants on what has been happening. So first, I'm showing you a 2015 photograph of members of the Knight Alzheimer's Disease Research Center. I'm seated here in the foreground here and flanked by Mr. Norman R. Say and Ida Goodwin-Woolfolk, who have been stalwart members of our African American Advisory Board. Unfortunately, since this photograph is taken, both Ida and Norman have died, and now the African American Advisory Board is led by Pastor Douglas Petty. The African American Advisory Board has been exceptionally helpful to us at the Knight Alzheimer's Disease Research Center in trying to learn how we can be more welcoming to people of color, both as research volunteers as well as faculty and staff. I'm very fortunate to be able to direct such a talented and large group of investigators and staff. Of course, I certainly can't do it alone. I'm assisted by my associate directors, David Holtzman, a noted Alzheimer's disease investigator and currently the chair of the Department of Neurology at Washington University, Randy Bateman, professor of neurology and principal investigator of the Diane study and the Diane Trials Unit, and now Carlos Kuchaga, who is a core leader, genetics and high throughput omics core of the Knight ADRC. These are associate directors. For almost 30 years, I benefited from the sage advice and support of Dr. Virginia Buckles as the executive director of the Knight Alzheimer's Center. She retired in June 2019, but had already groomed her successor, Dr. Krista Mulder, who now is executive director and is just as fabulous as Virginia has been in helping me to lead and to allow the Knight ADRC to succeed. I can't go through everyone here, but there are many, many people who make the Knight Alzheimer's Center as successful a program as it is, and I'm indebted to them all. But key to the Alzheimer's Disease Center and its Memory and Aging Project is you, our participants, and there are over 800 volunteers who, up until COVID-19, were coming every year to participate in their annual assessments and other studies. So without you, we would not be an Alzheimer's Disease Research Center. I mentioned this was taken in 2015. We had decided, because we can't do this every year, that we would take a group photo of the entire Knight ADRC every 5 years, and we're planning to do so in this past June 2020 when we originally scheduled this participants meeting, but of course, COVID-19 has thwarted those plans. So we'll devise another way to try to capture our current staff and faculty in the Knight Alzheimer's Center, as well as many of our friends. And among our friends, I would be remiss if I didn't remark upon Joanne Knight. The reason there is the Knight Alzheimer's Disease Research Center is in 2010, Joanne and her husband, Charles F. Knight, endowed the center and enabled it to reach its maximum level of efficiency and success that it has since achieved. So many thanks to Joanne. In the past year, we've had a number of additions to the Knight ADRC team. I'll talk a little bit about Dr. Joy Ballsbury, Dr. Kyle Womack in just a moment, as well as Dr. Jonathan Kipnis. But we've had a number of coordinators, schedulers, nurse clinicians, other staff join us, and we're very pleased to have all of these individuals as members of our team. Some people have moved on. Linda Amos, a longtime key member of our administrative staff, has retired. Anastasia Anazawano has left her psychometric position, testing position here at the Memory and Aging Project to go to medical school. And she's currently a first-year medical student at the University of Missouri in Columbia. At Columbia. These 5 individuals have gone on to other positions. Here are the new faculty members. I mentioned Dr. Ballsbury. She is a psychiatric epidemiologist and is very interested in health disparities and indeed had joined us as of July 1, 2020, coming from the Mayo Clinic in Rochester, Minnesota, to lead a new core in the Knight ADRC that focuses on health disparities and equity. Dr. Kyle Womack is a behavioral neurologist. For many years, was a key member of the Alzheimer's Center at the University of Texas Southwestern in Dallas, and he also joined us as of July 2020. And a neuroscientist, Jonathan Kipnis, came to us also in 2020 from the University of Virginia in Charlottesville, and he now is professor of pathology and immunology, and he runs a new center that looks at the role of inflammation, our body's defense mechanism against infection and other diseases, and how inflammation may interact with the brain. So we welcome all 3 of these new faculty members. After I provide this update, we're going to hear some individual presentations from the following individuals. Dr. Joyce Snyder is going to update us on the clinical trials component of the Knight Alzheimer's Disease Research Center. The trials evaluate experimental therapies that may benefit people who have Alzheimer's disease, and Dr. Snyder will talk more about that. And I was mentioning Dr. Kipnis and his interest in the role of inflammation in the brain. Dr. Bo Ansis is going to talk about a specific factor in our body's inflammatory mechanisms, that is the gut, the gastrointestinal system, and perhaps the role the gut and its regulation of inflammation may play in terms of Alzheimer's disease. Many of you have heard that a healthy lifestyle may reduce risk for Alzheimer's disease. Dr. Denise Head is going to propose a study looking at physical activity and exercise and whether it does reduce risk. I mentioned Dr. Ballsbury and her new core on health disparities and equity. Marissa Streitz is going to talk about capturing information that occurs over a lifetime that may influence whether we age successfully or whether we develop dementing illness such as Alzheimer's disease. These sorts of Life course factors include adverse childhood effects, perhaps divorce, perhaps poverty, lifelong discrimination, and the stress that accompanies that, and many other factors that may, as I say, influence whether people have risk for Alzheimer's disease. And then many of you have heard that we are interested in giving you, our participants, at least those of you who wish to know, results of some of the studies in which we ask you to participate, including your amyloid PET scans and your genetic profile. And Jessica Mazurski is going to talk about launching this as a matter of just a week or two, and perhaps by the time of November 14th, it will already be launched. So it's imminent. Well, what did the Knight ADRC do in response to COVID-19? Well, in the beginning of March, when we became aware of the impending threat of the pandemic, we began enhanced safety measures in our Memory and Aging project. And indeed, our last assessment was on March the 17th. And then from that point on, we, as the rest of the university and the Washington University School of Medicine, suspended all in-person assessments. So our Memory and Aging Project employees began working from home. I sent a letter to all of you showing my naivete about the seriousness or the duration of the pandemic, trying to explain to you why we have suspended our assessments. And I said, it's probably likely gonna last through April the 30th. Probably should have put April 30th, 2021. But I and many of the rest of us did not know how long this pandemic was going to last. In April, it became clear that the pandemic was going to disrupt in-person assessments for weeks or months to come. So we began telephone interviews to collect information. And we also began administering some of our memory and thinking tests remotely as well. And because COVID-19 can affect people in adverse ways, simply by remaining housebound and unable to interact with family and friends, we began a program where every Memory and Aging Project participant received a call from a staff member to assure whether, to find out whether people, how they are coping with this, and if they need help, who we might refer them to, including our social workers. So these are the programs we began in April. Since we began those assessments remotely, we've completed 345 telephone assessments and 251 memory and thinking testing sessions, and 472 of the shelter-in-place calls to assure the safety and well-being of our participants. This is terrific, and it's far better than not doing any assessments or any contacts, but nonetheless, COVID has had an adverse effect on our research. We would have normally expected to have completed by this time 480 assessments, whereas we only have 345, and 440 memory and thinking test sessions, where we have only 251. So we are playing catch up. But I have to say that all of our faculty and staff and all of you have been remarkable in devising ways that we can maintain our basic function and to stay in contact with and connected to you. Everyone has pitched in admirably and made a lot of progress in a very short time. We appreciate all of your study partners who we interview about you, our participants, to find out how you are functioning. And we thank all of our participants for agreeing to the cognitive testing remotely. We hope to return to, on a limited basis, at least certainly initially, to in-person assessments because many of the things that we do, including imaging, cannot be done remotely. but we only want to return to in-person assessments when it is safe to do so. We are developing plans. As a matter of fact, the entire university and the School of Medicine are developing plans to try to have a strategy wherein we can begin on a limited basis to return to in-person assessments, assuring the safety of the participants and the staff. Well, what is the Knight Alzheimer's Disease Research Center's mission? Well, we have a number of initiatives and goals that we are trying to address, from laboratory work to issues such as well-being and caregiving and driving, safety. So it's a range of initiatives that we study. But our major one is to look at the development of Alzheimer's disease in the brain prior to any indication of abnormal memory or abnormal thinking. That is, before dementia begins. And I think you've heard us say that we now have evidence that this stage where the brain lesions are building in the absence of dementia lasts decades, at least 20 years, perhaps longer. We identify those brain lesions through our imaging program, this brain MRI, magnetic resonance imaging, the PET scans for proteins, amyloid, and tau, and the concentrations of amyloid and tau in the cerebrospinal fluid. And hopefully, going forward, the identification of the concentrations of amyloid tau in the blood. And I'll talk about this at the very end. Our questions: does everyone who has evidence of brain lesions of Alzheimer's disease, but still remain perfectly normal in terms of memory and thinking, if they live, will they inevitably develop Alzheimer's dementia, symptoms of Alzheimer's disease? Is that universal, or will some people be protected against this progression? So that's one question. Second, can we identify when people who have preclinical Alzheimer's disease, the brain lesions, when they change from being normal in terms of memory and thinking to developing Alzheimer's dementia? Can we identify that period? And what are the factors that are important to transition from cognitive normality to Alzheimer's dementia? This is important because we aim to test drugs, some of the drugs I was mentioning in the clinical trials unit that Dr. Snyder will talk about, investigational drugs, as an effort not simply to treat people with Alzheimer's dementia, but prior to the onset of dementia, to prevent dementia. So you'll hear about that as well. And I've already mentioned that we're very interested in looking at the influence of factors that go with different racial groups and their role, a possible role in risk for Alzheimer's disease, social determinants of health. This is a scheme of the biomarker changes. I've put here the Alzheimer's dementia course, which usually lasts from onset, which is very mild, to severe, which is end stage Alzheimer's disease, lasts about 7 to 10 years. This stage only occurs because those brain lesions that have been building up for 20 plus years ultimately cause brain cells to die. Brain cells are called neurons. So once neuronal loss occurs, the symptoms of Alzheimer's disease occur. Many of you know that there have been many trials of experimental drugs to try to treat Alzheimer's dementia. And to date, they've all seemingly failed. In part, we think, because by the time you have Alzheimer's dementia, it means your brain cells, or many of them, have been badly injured or are dead. So we're treating an irreversibly damaged brain if we wait until the dementia stage. Plus, it's not just brain cell loss. There are many other factors that are going on in the Alzheimer's dementia brain, and we can't just treat one aspect. We might need to treat multiple aspects to be successful. But our interest is in this preclinical stage of Alzheimer's, when the memory and thinking is preserved, but gradually we're building up the brain lesions of Alzheimer's disease that we can detect through examination of the cerebrospinal fluid, PET imaging, and try to intervene somewhere here with these investigational drugs to prevent the progression to Alzheimer's dementia. Just to clarify some of the terminology, I've used the name, the Memory and Aging Project. It is a key part of the Knight Alzheimer's Disease Research Center because this is where we do the in-person evaluation, interviewing you and your study partner and testing you on memory and thinking. So the Memory and Aging Project is the clinical office where we do this and the faculty and staff that are involved in this. This was the oldest part of the Knight Alzheimer's Disease Research Center, actually began 6 years before the Alzheimer's Center was formally established, began in 1979 under the leadership of my mentor, Dr. Leonard Berg. So this has been going on for over 40 years. The Alzheimer's Disease Research Center was given a large award by the National Institute on Aging, part of the National Institutes of Health, beginning in 1985. And I've mentioned that in 2010, Charles and Joe and Knight endowed the Alzheimer's Center. So since then, we call it the Knight ADRC. But this grant for the ADRC is not the only grant that supports our research. We have two other large grants. We have many grants, but two other very large grants, Healthy Aging and Senile Dementia, and the Adult Children's Study. I'll talk a little bit more, particularly about the Adult Children's Study. No matter how an individual volunteer is supported under which grant mechanism, all assessments take place in the Memory and Aging Project Research Office, MAP. And then in 2008, we added a very special group of volunteers, those who have a rare mutation that causes Alzheimer's disease called the Dominantly Inherited Alzheimer Network. We're very fortunate that we are supported by many partners and collaborators, certainly the Greater Missouri chapter of the Alzheimer's Association. I mentioned the fantastic support we get from our African American Advisory Board. We have an internal ethics committee on which some of our participants are key members. The Links Incorporated, the St. Louis chapter in particular, the Harvey A. Friedman Center for Aging at Washington University, the Hope Center for Neurologic Disorders, the Barnes Jewish Hospital Foundation, and Washington University, including from Chancellor Andrew Martin, the Dean of the School of Medicine, David Perlmutter, and I mentioned our current department chair, David Holtzman. Our formerly the Alumni and Development Office, or now the Advancement Office, has these individuals who all support us with our donors and our friends, including the Knight family. I mentioned them already, and I'll come back and talk about Roger and Paula Riney in just a moment, but many others who are very generous supporters of our work. So what about you, the participants, you as a group? Well, right now, there are 822 of you. Some of the grants that we have, the Healthy Aging and the Alzheimer's Center grant, enroll people who are 65 and older, and there are 526 of you in that age category, where the Adult Children's Study enrolls people 45 and older, and there are almost 300 of the adult children. So the age range of the people we evaluate goes from the 5th through the 10th decade of life, age 45 to 96 years. Slightly more women than men, 16% are African American. One of you has participated every year for 33 years. That's our longest current period of participation, and another person has done so for 30 consecutive years. Remarkable. And of course, the vast majority of you live in Missouri or Illinois, but some come from many other states, including Idaho, Texas, California, and Maine. On average, those of you who are cognitively normal, no memory and thinking problem, are about 76 years of age. We have two categories of very early Alzheimer dementia, designated by 0.5 and 1, and these people are 78 and 75 years on average. All of you are remarkable. I just mentioned Miss Carlene Davis, who recently was designated as a Woman of Achievement in this past year, 2020, for her lifetime achievement efforts as a public school teacher and then an after-school volunteer. She's been a Memory and Aging Project participant since 2004. We are very fortunate. I mentioned at the outset, we have such a remarkable group of faculty and staff, and you are participants who support us with your time and effort. And we are rewarded for this when we apply for funding to support our research. We get not quite $200 million a year, this past year in 2020, to support our research. This is not simply the Knight Alzheimer's Disease Research Center. It's for all Alzheimer research at Washington University, but that's a remarkable amount. And it put it into perspective, our sister institutions in Kansas and Missouri and Illinois get maybe 1% of that, 2 million for University of Missouri, St. Louis University, maybe 10% of that for the entire University of Illinois system and the University of Kansas system. So we are remarkably fortunate with such talented investigators and staff and such committed participants. And this is not something that is recognized only locally, but we get lots of requests for the high quality data that you help us generate. Now, every 5 years, our individual grant awards from the National Institutes of Health, we have to reapply for another 5 years, the next budget period. And we just did so for our adult children study, submitted back in May in the midst of the pandemic, an application that if it is awarded, will begin June 1, 2021 and go through 2026. We're requesting in that about $22 million for that grant alone. The application was over 750 pages. But this, as I say, is just a manifestation of how valuable the data that you generate has become. We get, over the past 5 years, requests from all over the world, certainly here at Washington University, but all over the world, to have access to the data that you generate and the tissue samples, including your spinal fluid and your plasma, so that research can be supported throughout the world with your own contributions. So you are certainly integral to our research goals, but your participation really benefits global Alzheimer research. And remarkable, it supported about 273 manuscripts published in highly read peer-reviewed journals to advance understanding of Alzheimer's disease. You have made a remarkable contribution to the understanding of Alzheimer's disease, not only at Washington University, but worldwide. We get other centers asking us how we can do this so well. They seek our input and guidance. Just mention that before our adult children's study was officially funded in 2005, we did a small pilot study of middle-aged people who had no memory and thinking problem, but had a parent who had Alzheimer's disease. Would they participate in research? Would they take time off from work and time off from family to come in and undergo a lumbar puncture, a spinal tap to get cerebral spinal fluid, or imaging studies? Because we didn't know whether this was feasible or not. We signed up 93 original people for this pilot study, and 44 of them, almost half, continue today 18 years later in our adult children study. Again, remarkable testament to the dedication and commitment of our participants. We cannot do our biomarker studies, which is how we characterize preclinical Alzheimer's disease. The brain changes are beginning in the absence of any memory or thinking problem. We cannot do that until we restart in-person assessments. And so these biomarker assessments are critical for the continued research advances we wish to make. And it's not simply a one-time assessment. We have to study change over time. Let me illustrate this with a potential biomarker study of the brain protein amyloid, which we think is critical to Alzheimer's disease. So for the first time, this is hypothetical, a person was tested, their amyloid level was 3. Then a year later, their amyloid level was 2. Now, no test is 100% perfect. So maybe this lower test was abnormally low because of a test problem, or maybe this original test was abnormally high because of a test problem, or maybe there truly is a loss of amyloid protein over time. How do we know this with following somebody only twice, year 1 and year 2? Well, let's see what happens, all hypothetical. At year 3, if the amyloid level is still getting lower, down to 1, well, then we are more confident that whatever is going on is a real trend, a real observation. Maybe somebody had a drug that is reducing amyloid, and we can detect this not by simply 2 time points, but by 3 time points. Or maybe we find at year 3 that this is the level again, so we suspect that maybe this one was an incorrect one. Or at time 3, we find that the level is here, so now we suspect that this was accurate and this was the abnormal test. So I cannot stress enough how multiple times of testing is crucial to our ability to understand what is going on in the brain and how we can best address it. So I take my hat off to all of you who undergo multiple biomarker assessments, imaging, spinal fluid, blood. It is absolutely critical to our ability to decipher the process of Alzheimer's disease, and is why, because you do it at such a great rate, it is why people all over the world seek your data and your specimens. It is unique. So I just show you for our adult children study participants how many of you agreed to and completed your brain MRI scan. At time 1, 93%, 3 years later, 100%, 6 years later, 100%, 9 years later, 91%, and 12 years later, still 75%. Remarkable. Of course, the numbers who are eligible for this keep going down because not all of you have been enrolled for 12 years. But of those who are eligible, it's remarkable. And the same with amyloid PET. A remarkable agreement to have longitudinal testing, which is, as I said, is key to understanding Alzheimer's. Even the spinal tap for cerebral spinal fluid is in the 80s through year 6, down about to 2/3 at year 9, and about half at year 4. So thank you, but I wanted to explain why we ask you to do this, and why we are so appreciative of the fact that so many of you do. It is unparalleled information to see change over time in these biomarkers. We appreciate everyone who agrees to donate their brain after death. I promise you, we will not ask you to donate your brain as long as you still are using it, but after death, if you would donate it for brain autopsy, that still remains the gold standard for understanding Alzheimer's disease brain pathology. About 77% of you agreed to do that, including those of you who have no dementia. About 2/3, or not quite 2/3, do that, and about half of our African Americans. Contrast this with the autopsy rate for all 32 Alzheimer's centers throughout the country. General autopsy rate is about 64%, we're at 77%, and for African Americans about 25%, we're at 54%. So I mentioned how fortunate we are to have our research funded, and we are, but frankly, it covers only about 70% of our operational costs. So we're very dependent upon philanthropy, and we have been extremely fortunate to be the beneficiaries of remarkable philanthropy, including 2019, additional gift from Joanne Knight and her family, and from Roger, and a new gift from Roger and Paula Riney. This enables us to establish the clinical trials unit that you'll hear about from Dr. Snyder, to improve our neuropathology core with new equipment, and our clinical core where we assess people. And another recently received gift from a former patient of mine is also very much appreciated. I mentioned how worldwide people approach us to try to have access to your data and your samples. And here are some of the studies worldwide that seek us to collaborate with them because our data set is so valuable. We have other collaborations with other institutions across the United States, across the world, really, including about early onset Alzheimer's disease. This is when dementia occurs below the age of 65. A large neuroimaging initiative, Down syndrome. Some of you may know that all individuals who have Down syndrome and live to age 40 all develop Alzheimer's disease. And so this is another way to understand the development of Alzheimer's disease in another group of individuals. This is a non-Alzheimer dementia, frontotemporal lobar degeneration, or frontotemporal dementia. We participate in that as well. We are very interested in understanding whether in-person assessments always will be required, or whether we can utilize technology to obtain the information we want. So we're looking at ways for online assessments where our participants can self-administer a test at home on their smartphone or their laptop. And so we're working with another group to try to evaluate this. Of course, to know whether the online system is as good as or better than our in-person assessments, we have to do both, and for at least a period of time, and compare which one seems to be superior to the other. Dr. Jason Hassenstab in our psychometric group has also launched a study of testing individuals, not here at the Memory and Aging Project, not in person, but in normal settings, at home, at leisure activities, for a brief smartphone assessment, about 5 minutes or less. For a week, you may be asked to take a less than 5-minute test 4 times a day, and then Dr. Hassenstab and his team can evaluate whether this is better than having you come in to the Memory and Aging Project for the psychometric testing. So frequent testing, very short, look at the combined results. He started out with 144 participants, but it's going to add new participants each week. And most of the parameters about whether it was acceptable or not from the user experience seems to be either neutral or positive. Privacy concerns, which he has clearly addressed, voiced by some of you, and getting the program installed was difficult for some, but mostly it's been very well accepted. If we can do these short tests, then we won't need so many of you to come in for the in-person psychometric testing. We're always looking for your feedback so that we can optimize the test experience. We find that our African-American participants on some different test measures have different values than our white participants. Now, whether this, and particularly we find this in some of the proteins we evaluate in their cerebrospinal fluid. Question is, do we have enough African-American participants to be able to make a firm conclusion that what we observe is truly accurate, or whether it's an idea that with more data we would not consider. So we always are interested in improving the recruitment and participation of African-American participants in the new Health Disparities and Equity Core led by Dr. Balls-Berry is charged with helping us with this. But also we're interested in whether some of the factors that I mentioned earlier, social determinants of health, discrimination, stress, living in a neighborhood that it has adverse experiences, crime, lack of access to fresh fruit and groceries may be underlying whatever we observe as changes. So we're looking at this as factors to potentially explain racial differences. And our head of our biostatistics and data management core, Kenji Zhang, has a new grant from the National Institute on Aging to explore this in collaboration with two other Alzheimer's centers, one at Emory University in Atlanta, the other in Philadelphia, the University of Pennsylvania. Then I just want to point out that if we can find a more convenient and accessible and less expensive and invasive way to assess Alzheimer biomarkers, by all means, we're interested in that. These are individuals down here who had an amyloid PET scan. 115 of the individuals with an amyloid PET scan had no amyloid, but 43 did have amyloid. And in fact, of people age 70 and older, we find that almost a third, 30% or so, of normal 70-plus-year-olds will have an amyloid PET scan that is positive, preclinical Alzheimer's disease. This is not a rarity. It happens in a fair proportion of healthy older adults. Well, looking at the people who were at a positive amyloid PET scan versus a negative amyloid PET scan, Dr. Suzanne Schindler and her colleagues in the Bateman laboratory assessed the amyloid protein in the blood, in the plasma, a component of the blood. And they found that those individuals who had a positive amyloid PET scan had a low amount of blood amyloid compared to those who had a negative PET scan. Their blood amyloid was higher. In other words, it looks like low plasma amyloid test may be a way to indicate who has amyloid in the brain, which, if it's true, may mean we don't have to do an amyloid test, amyloid PET scan, or maybe even a cerebrospinal fluid. So this is very exciting new work that we're very much investing in to see if this is one way to have a safe and accessible, cheap, non-invasive screening test to see who may be amyloid positive and who may be not by assessing the blood. Well, every year at holiday time, we have a holiday card that we send to friends and colleagues throughout the world. These typically, for some reason, feature me as the director of the Knight Alzheimer Center. So once we had me at the Arch, once during a snowstorm when the university didn't clear our sidewalks, we had me out shoveling the snow so our participants could safely get into the Memory and Aging Project for their in-person assessments. 2018, we had in August a solar eclipse, and so we had me sort of simulating that I'm eclipsing Alzheimer research. And then last year, of course, when the Blues won the Stanley Cup, we had me holding the Stanley Cup and just wondering where I'm going to be able to display it here in my office. As I look back at these holiday cards, I notice that they all feature me. And I think we have grown so large that it may not be appropriate to have just a single leader for all of the different projects, initiatives, grants that we are doing. Moreover, I am 72 years of age. I once again indicate I have no definite plans for retirement, but of course I won't be here as the director forever. So we do have to think of getting people in place to be able to take over as director when I do decide to retire, if I'm unable to continue. And so I've been thinking about this, and some of you are aware that I have a grandson, Jack, who now is 5, almost 6, and I've been grooming him to be able to succeed me in the future once he gets a little bit older. But the fact that we are so big now that probably we'll need more than just one person to lead the program makes me think that perhaps I ought to bring on, in addition to my grandson Jack, my grandson Thomas, who is 3, and perhaps they eventually will succeed me as co-directors of the Knight Alzheimer's Disease Research Center. By the way, here's my lovely wife, Lucy, who is key to all that I'm able to do. She's a tremendous partner in this. So with that, I will thank you for your attention and look forward to interacting with you again on November the 14th when we have our panel discussion. So thank you very much.
Hello, this is Joy Snyder. I'm a clinician at the Memory and Aging Project of the Knight ADRC, and I also direct the Knight ADRC Clinical Trials Unit. Today, I'm going to give you an overview on clinical trials, both at the Knight ADRC and around the country. It has been a very exciting year and challenging year for clinical trials in Alzheimer's disease, both worldwide and particularly for us here at Washington University and the Knight ADRC. The exciting part is that for the first time in 17 years, the FDA is reviewing a new drug application for an Alzheimer's disease therapy. This is pretty exciting, and I'll tell you more about it in a minute. The other challenge has been that clinical trials here at WashU and worldwide have had to learn how to deal and operate with the COVID pandemic. This has been a challenge, but we are still conducting clinical trials and will continue to do so because we think this work is critically important. And finally, the Knight ADRC Clinical Trials Unit was able to move into our new space in October of 2020. Also an exciting time, but a little bit more challenging because of the COVID pandemic. The biggest question in clinical trials right now is, will we have a new treatment for Alzheimer's disease in 2021? The treatment that is under consideration by the FDA is a medication called aducanumab. It's a drug that's given intravenously in your veins, and it is a monoclonal antibody that binds to beta amyloid, that main component of the plaques in Alzheimer's disease, and may help clear those plaques out of the brain. A company named Biogen conducted the studies of this drug, and they conducted 2 studies called Emerge and Engage, which were identical but were separate so that they could have 2 separate studies to analyze. These studies enrolled over 3,000 participants worldwide, including 10 participants here at the Knight ADRC. These folks had mild symptomatic Alzheimer's disease and received monthly infusions of aducanumab or placebo. The original goal of the study was to treat people for 18 months. The study drug dosage was increased during the study because they decided it wasn't high enough at the beginning. But then several years into the study, they conducted an interim analysis, looked at some of the data, and concluded that the drug probably wouldn't work. So they stopped the study in April 2019. However, a few months after that, they reanalyzed the data, mostly for the people with the higher dose of drug, and said, maybe this drug did work. So after that reanalysis in July 2020, they asked the FDA to approve the drug, and this is currently under review. They expect the response from the FDA in early to mid 2021. This is complicated because they didn't complete the entire study, and only one of those 2 identical studies showed a significant benefit. All the folks who were in those studies have now been offered an open label extension in a study called Embark, and 7 participants here at WashU will receive this treatment. I just showed at the bottom of this slide some of the data that's available from those studies, and this is using a measure called the CDR sum of boxes, which is a test that was developed here at WashU, and a higher score means your memory's worse. So over the course of this study, which went about 1.5 years, 78 weeks, the people on placebo did slowly get worse. The people on the 2 doses of the medication got somewhat less worse. So the study drug did help slow down progression of the disease. But this is results only from eMERGE, which is one of those studies. The other study showed a similar trend, but it wasn't significant. The more exciting result was looking at the amyloid in the brains of the people who were treated with this drug. So this is looking at amyloid PET scans using a measure of how much amyloid is in the brain. And for the people on placebo, there really wasn't much change over the year and a half of the study. They stayed about the same. But the people on the two different doses of medication had a significant decrease in beta amyloid. And on the higher dose, these people are almost down to what would be considered the negative range, what we'd see in someone who didn't have Alzheimer's disease. So this is pretty exciting, but the data is incomplete. We only have it for a very small number of people. This N number is the number of people who actually had this result, and it's less than 80 people out of 3,000 people in the study. So really incomplete data. So what will the FDA decide? We don't know. But if they do approve the drug, it will have some big implications for life going forward for all of us. There are a lot of other things going on besides that one particular study, though. At the Knight ADRC, we are doing 3 other studies using amyloid antibodies to try to reduce amyloid plaque and slow symptoms in Alzheimer's disease. And I've listed those for you here. These are studies sponsored by Eli Lilly for Trailblazer, by Eisai for Clarity, and by Roche for Graduate. These are all underway, and we have people here at WashU getting these study drug infusions. These study drugs will be completed in a couple of years, so we won't know until 2021 or 2022 whether these drugs might be promising and go on for FDA approval. And then the final study there is the EMBARK study, which is the open label study of aducanumab. We also have other studies going on at WashU. You've heard probably in the past about the DIAN-TU, the Dominantly Inherited Alzheimer's Network Treatment Unit. These are studies for people who come from a family that have a mutation that causes early onset Alzheimer's disease, often before the age of 60. It's an international study with 20 active participants here at Washington University. 11 of those participants are currently being treated with gantenerumab, another one of these monoclonal antibodies. That study completed and showed some promising biomarker effects. So those 11 people are now in what's called the open label extension. They're all getting the study medication. 9 people are in a study we call the cognitive run-in that will be ready to have new drugs as new drug arms roll out, probably in 2021. We're also in the startup phase for 2 new studies that will be for people with symptomatic Alzheimer's disease. One of these is called Green Memory. It's a study of a drug that was recently approved in China. So this is an oral medication that may help people with more severe symptoms of Alzheimer's disease. And the other study is called Autonomy, which is a study of an antibody against tau rather than beta amyloid. So this is very exciting, as this is the first tau therapy we've been able to study here at Washington University. So we're excited about those studies coming online later this year and in early 2021. We also have studies of people who do not yet have symptoms of Alzheimer's disease. This is what's called asymptomatic or pre-symptomatic Alzheimer's disease. You've heard about this at these meetings before. We can now tell if someone has the brain changes of Alzheimer's disease using the amyloid PET scans or spinal fluid markers, and maybe soon even using blood markers. So these studies include the A4 study, which is anti-amyloid treatment in asymptomatic Alzheimer's. Don't try to say that fast. This study started here at the Knight ADRC in October 2014. There are over 2,200 people randomized around the country, and we have 11 currently active here at WashU. Enrollment in this trial is completed, and 5 of our 11 participants are now in the open label extension part of this trial. Other people around the country and here at WashU are completing the study period, and we expect results from this study in 2022. This will be very exciting as one of the first studies in sporadic pre-symptomatic Alzheimer's disease to have results. Ahead is the next version of that study. It's a study of people who are at risk for Alzheimer's disease but do not yet have symptoms, and it will use a different one of those monoclonal antibodies to treat people. This will treat people who have very clear amyloid buildup in the brain and those who are in sort of a borderline zone, so extending the treatment even earlier. Just like that A4 study, we will need to screen a lot of people in order to get people who have the right amount of amyloid to be in the study. So we had to screen 73 to get 15 in for the A4 study, and it'll be similar for AHEAD. One of the big focuses of AHEAD is to work very hard to include minorities in recruiting. So we will be doing that here at WashU because we want these studies to be inclusive and to help us learn how to treat Alzheimer's disease in everybody. The last study is a different one for us. It's a study that's strictly observational. It's called TrackPad. This study is so we can bring people in and get them ready to be into a clinical drug trial study before we are ready to start the drug trial study. So it can be used as a launch pad for future interventional studies. So the future of clinical trials at the Knight ADRC is really bright. I'm very excited about the group we've developed and all the studies we're being able to do. This is much more than we could do in the past. We do have the world's best participants, and we do have the world's best staff and the world's best ADRC leadership, as you all probably know. But that's not enough. So we need more staff to keep going and increasing the number of clinical trials we're doing. And we need more participants. So we're working on bringing in more participants, both to the Knight ADRC studies, including the Memory and Aging Project and the adult children studies, and for clinical trials. So tell your friends, we need more people to help us in the fight against Alzheimer's. We did open up our new space recently. I've got a picture here of the construction ongoing, and then this is the finished space. And as of last week, we even have furniture. So we are ready to go in our new trials unit space. We hope to have an open house and show everyone. We have had some changes in our staff that I did want to mention. We promoted Sonia Siemens from regulatory coordinator to project manager. Sonia's here. We promoted Amy Evans from coordinator to be the regulatory coordinator. Jennifer Fisher-Estep, who some of you may recognize from her time at the Memory and Aging Project, is now our full-time lead psychometrician. Marta Santos, whom you also may recognize from Memory and Aging, has moved full-time to the Knight Clinical Trials Unit. She's helping us with the A4 study and with the AHEAD study. Dr. Kyle Womack recently joined the Knight ADRC, and he will serve as the Associate Director of the Trials Unit. And Sonia Stocking just moved from our Memory Diagnostic Center clinical practice to join us as a full-time psychometrician. So lots of exciting staff changes. The bottom line for clinical trials is that we may or may not have a new drug for symptomatic Alzheimer's disease dementia in 2021. That's up to the FDA. If that drug is approved, it means we will need to increase our capacity here at WashU and everywhere to screen and diagnose Alzheimer's disease, including having more availability of imaging with PET scans and more availability for infusions. Whether it's approved or not, the somewhat positive results will promote continued development of Alzheimer's disease drugs, and that's good news for everybody. The ongoing trials will tell us if we can slow down Alzheimer's disease with disease-modifying drugs, drugs that attack the underlying disease, and if we can stop the disease before it starts in the pre-symptomatic people. So that's very exciting as well. And all of that has been made possible because of the work that you have done by participating in the studies at the Knight ADRC. All that spinal fluid you've given us, all those PET scans, all that memory testing you put up with, all those questions at the clinical assessments, those things are what gave us the data that enable us to know how these biomarkers predict Alzheimer's disease and how we can attack the disease. So you really all need to pat yourselves on the back for everything that you have done to promote the search for a cure for Alzheimer's disease. I do want to highlight one of our participants, Dr. Collins Lewis, who is a former colleague here at WashU and also a participant in the Memory and Aging Project, and has served on our African American Advisory Board and also served as a volunteer in the A4 study. Dr. Lewis was recently honored as a recipient of the Cornerstone Award from the Global Alzheimer Platform Citizen Scientist Award. All of you are citizen scientists like Dr. Lewis, and all of you can help us fight Alzheimer's disease and end up with a world without Alzheimer's disease. So again, thank you very much. I hope you enjoyed the participant meeting presentations, and I look forward to seeing you via Zoom at the Q&A session. And I also look forward to the day we can all see each other again in person at the Knight ADRC and at the Memory and Aging Project. Thanks.
Welcome. My name is Bo Ansis. I'm a neurologist here at the Knight Alzheimer's Disease Research Center. And my area of interest is looking at early biomarkers for the diagnosis of Alzheimer's disease. Today, we'll be looking at the role of the gut microbiome in Alzheimer's disease. I have no disclosures for this talk. There are a large number of microbial communities throughout our daily interactions. And these communities are highly diverse, abundant, and serve several important functions. So there is the microbiome that we see in our gut. There is also a biome that we see on our skin, or even in our mouth, or on our closest friends and pets that we may see. And these biomes are present not only at certain ages, but can be even seen in babies, and they are directly influenced actually by the mother. Now, these biomes give us a window into understanding both health as well as disease. In particular, the gut microbiome is very important because the gut microbiome promotes healthy gut lining. And when there are changes in this biome, they can affect our immune system, and those same changes can also affect inflammation. Now, this biome is also very important for it allows us to absorb certain kinds of nutrients from our gut. In particular, it assists in the synthesis of vitamins and nutrients. And it also provides important information to the rest of the body. In fact, there is actually a gut-brain axis that actually exists within individuals, such as you are really who you are, what you eat. In particular, the changes that we see in our gut microbiome affect the brain. Those changes in the brain then change or affect our gut. And so these kinds of changes can be seen even if we're at different times of the day, because if we're hungry, there are changes within the gut that then affect the brain. Those changes in the brain will then affect our gut. So we're very interested in looking at this biome for understanding what is happening in different diseases. Well, what do we know about Alzheimer's disease? We know that Alzheimer's disease is a slowly progressive process. And now, using certain criteria, many of which have been well developed and actually been a leader in has been WashU for characterizing these kinds of changes that are seen in Alzheimer's disease. In particular, the first changes that we typically see are changes in amyloid. Those changes in amyloid often occur 15 to 20 years before an individual becomes symptomatic with the disease. These changes in amyloid are then followed by changes in tau, with individuals changing from normal to abnormal or higher levels or deposition of tau within the brain. After that, there are changes in brain volumetrics, or the brain structure, that we can easily image using magnetic resonance imaging. Now, later on, we'll often see changes in cognition, but those changes on those pen and paper tests usually occur only a couple years before an individual shows true symptomatic signs or changes that other family members or that individual may recognize. So using this criteria, we've been able to stage an individual by this A for amyloid, T for tau, or N for neurodegeneration. While these markers are very important and allow us to understand some of the pathology or the changes that we see within the brain, there are other measures that could also be very important. We've been very interested in looking at the gut, especially as we said before, because of the strong gut-brain axis, and just try to see if there are changes in the gut that are associated with these particular biomarkers of the disease, such as that we may be able to stage when do the changes in the gut occur. Do they occur soon after amyloid, soon after tau, or soon after neurodegeneration? So the goal of our study is to characterize this gut microbiome composition as well as the function in both healthy individuals, those that are healthy but who have preclinical Alzheimer's disease, meaning have changes in some of amyloid and tau, or those individuals who have symptomatic, who have both thinking issues as well as changes in amyloid, tau, or neurodegeneration. We are looking to evaluate individuals both cross-sectionally, meaning at one time point, as well as longitudinally, repeatedly following these kinds of individuals. So how do we do that? Well, we actually send home stool samples, and we have a stool sample collection method. So study staff provide a stool kit, which is sent to the home, and we also provide a food log questionnaire. When the individual is going to have a bowel movement, they complete and provide that sample at home, as well as they complete a series of questionnaires of what their most recent or last 24 hours of food intake. A courier service actually comes to the home, picks up the stool samples as well as the questionnaires, and then they are processed and sent here to WashU. So it is a very, very easy and very, especially easy now and even in the times of COVID, a method for obtaining these samples within individuals. And so we are asking individuals and participants to provide a stool sample both annually to look at these kinds of changes so that we'll be able to see both cross-sectionally and longitudinally changes in the gut microbiome as a function of clinical status. So if an individual is cognitively normal, preclinical, or symptomatic Alzheimer's disease. So our experimental design is to look at individuals who are both healthy, preclinical AD and symptomatic AD, do, again, this longitudinal sample where we're following individuals. And in some cases, some of these individuals may change from healthy to preclinical, or some of our preclinical may change to symptomatic. And we're following this conversion as well as looking at individuals longitudinally. When individuals are often coming in for their cognitive assessment, we're asking them if they're also willing to provide a stool sample at home. We send those stool kits to them, and then we collect the gut microbiome, as we mentioned before, by courier service. Once we take the stool sample, we extract the DNA, and we get what is the microbiome, or what are the different organisms, as well as how much of a particular organism is present. We do this through DNA sequencing, as well as data analysis. And this allows us to understand, are there certain microbes and microbial activities in the gut and how they relate to the stage an individual is in regards to Alzheimer's disease. So using these kinds of methods, we've been able to look at individuals and actually classify them according to their stage and trying to see if there are differences in the microbiome. What we've overall seen is that actually there is. And here on the left-hand side, I show you a plot of individuals who are healthy, which are in red, or in blue, those are individuals that have preclinical Alzheimer's disease. Now we use some fancy methods and statistical analyses to try to separate out these different groups. And this is done by using simple methods of looking at the diversity of these different microbiomes within these two samples. And what you start to see is that there seems to be two separate circles. So one for those with preclinical that seem to kind of clump together, and two for those that are healthy. Well, we then looked at those relationships in regards to those other biomarkers that we're also collecting on in individuals, in particular in regards to this A for amyloid, T for tau, or N for neurodegeneration. Now, how do we get those markers? Well, for amyloid, we use something called positron emission tomography, or PET imaging, where we stick a small radioisotope into an individual. It's very safe and very easy. They lie quietly in the scanner, and then we wait a period of time and see if there's an increase in uptake in the amyloid. For tau, we do the same kind of, again, using this PET technique with a radio tracer that has high affinity or has high ability to bind to tau within individuals. And we look at if there is increase in tau uptake within individuals. And then for cortical signature or for neurodegeneration, we measure the brain volume. So we take a simple MRI picture, and that allows us to quantify how much of the brain or how big certain areas of the brain are. And what you can see is on our x-axis is this diversity scale, and on the y-axis is the uptake either with PET imaging for amyloid, for tau, or for cortical signature or for brain volume. And what you can see is that overall our healthy individuals have low amounts of amyloid compared to our individuals who have preclinical Alzheimer's disease who have higher amounts of amyloid. And these same individuals are preclinical also have smaller amounts of tau compared to a little bit higher amounts of tau within our preclinical Alzheimer's disease. And our cortical volumes were pretty similar for all of these groups. So what we did is we tried to compare, is there any kind of relationship between this diversity that we see for individuals as a function of these different measures? And what you can start to see is there does seem to be a strong correlation with this in regards to amyloid compared to either tau or neurodegeneration. So that would suggest to us is that there are already starting to be changes in the gut microbiome, or so those different kinds of species that are present that are changing when an individual starts to become amyloid positive. So they may be cognitively normal, but there are already changes in amyloid deposition in the brain. And those changes in amyloid deposition are also affecting our gut or brain diversity that is present. Well, it's not, as I said to you before, looking at the gut microbiome, we can look at the diversity, but we can also look at the function of those different organisms and what are they involved in. And so we've started to look at what are those different organisms and what are they doing. And we can see that in certain cases, in those that have preclinical Alzheimer's disease, again, we're looking at the two groups as a function of a different amount of a species of a particular organism. There are higher amounts of certain organisms, and these are very, and my Latin is not as good, but we'll try to go through them. Ruminococcus bromai or Eubacterium ventriosum. So there seems to be higher amounts of these certain kinds of organisms that are present in preclinical Alzheimer's disease. But in other instances, there's actually a drop in other organisms. So there is this kind of yin-yang relationship that we're starting to see. So there is actually a drop in E. coli or Biophila unclassified, where there seems to be drops in these kinds of organisms in preclinical. And so it's this differences in this milieu that may be important in the gut microbiome, and that may also affect the function of the gut microbiome. And so here are a long list of different pathways that we see and their association with certain kinds of those organisms. And so this is looking at metabolism, so how well the brain cells or other parts of the body function. We can also look at the cell wall. We can look at how vitamins are being absorbed. We can look at how amino acids, those are the basic building blocks that make up proteins of who we are, as well as even how sugars are being regulated within these individuals. And what you see in green are changes that are associated with more with preclinical status, and in red are changes that are associated in the healthy. And in healthy, if there's a negative, that means there's a drop. And in green, if it's a positive, if you're seeing that, then that means an increase in them. And so what you're starting to see is that there are changes in how the cell walls are functioning, how vitamins are being absorbed in preclinical compared to healthy status, as well as even how amino acids are being made or proteins that are being made. And so these processes can then help us understand how these can possibly lead to changes in amyloid deposition as well as tau. And so our future studies are to look at these kinds of pathways in a much larger group of individuals who we hope to look at, again, both cross-sectionally and longitudinally. So in summary, what we've tried to start to show you is that there are changes in the gut microbiome. So what makes us up in our gut is changing, and those changes seem to be early on in the disease process, in the preclinical stages, in particular with amyloid. But I wouldn't be able to do this without the great team that we have here at the Knight ADRC and specific investigators, in particular, Dr. Susie Stark, who you've heard from in the past, and two gut doctors, Drs. Phil Tarr and Drs. Gautam Dantas, and then our teams that are helping on the collection of that, Becky Bollinger and Regina Thompson, as well as great students that are working to study these gut changes, Aurora Ferriero, Juhi Choi, and Aaron Newcomer. I wanted, again, thank you for participating today and coming to these lectures, and I look forward to your questions. Thank you.
Hello, I'm glad you're able to participate in this opportunity for us to show our appreciation for your contributions to our research effort. I'd like to take a few minutes to share some planned research on physical activity and successful aging. When I think about successful aging, I like to remember the story of Ernestine Shepherd. She's a personal trainer and competitive bodybuilder. As a 56-year-old, she was a sedentary school secretary who had never worked out a day in her life, really, until one day she decided it was time to get in better shape. Starting slowly and building her body step by step, Ernestine over time completely transformed not only her body, but her life. In 2007, at age 71, she began to train to compete as a bodybuilder. And in March 2010, she was given the title of the world's oldest performing female bodybuilder. Stories like this are a driving force for my research on looking at factors that may help promote such successful aging for all of us. So physical activity and exercise may influence aging and Alzheimer's disease. Physical activity refers to any energy expenditure derived from any bodily movement produced by skeletal muscles, while exercise representing a subset of physical activity that involves the goal to increase or maintain physical fitness through structured, repetitive bodily movements. We all know that exercising is good for our physical health, including our heart and lungs, but accumulating evidence also suggests that exercise is good for our brains and thus our memory and thinking skills. Importantly, there's also some evidence that physical activity and exercise may reduce the risk of developing Alzheimer's disease. For example, in this study by Aaron Buttman and colleagues in 2012, the researchers assessed for the presence of Alzheimer's disease over 5 years in individuals who were either low or high in physical activity. They observed that those low in physical activity had a greater risk of developing Alzheimer's disease than those high in physical activity. While some of the existing research is promising, one key question is, how might physical activity reduce the risk of Alzheimer's disease? We are planning a research program to address this question. Our goal is to examine a specific model suggested by animal studies on how physical activity may be beneficial for brain health and memory. First, we will robustly measure physical activity and exercise in 3 ways. One measure will be cardiorespiratory fitness, which reflects the level at which your heart, lung, and muscles work together when you're exercising for an extended period of time. This shows how efficiently your cardiorespiratory system functions and is an indicator of how physically fit and healthy you are. Cardiorespiratory fitness is measured as the maximum amount of oxygen that the body is capable of using during high intensity activity. We will estimate cardiorespiratory fitness by having individuals cycle on an exercise bike at varying intensities while their heart rate is measured. Then an equation is used that incorporates age, sex, and aspects of heart rate to estimate maximum oxygen uptake. Next, actigraphy allows for objective, unobtrusive measurement of activity levels over extended periods in the home and during normal activities. The technology uses a small watch-like device to continuously monitor movement. In our study, the actigraphy device will be worn on the hip for 7 days to obtain estimates of engagement in moderate to vigorous physical activity. Lastly, we will obtain a self-report measure with questions about the time being spent physically active over the last 7 days, including job, transportation, household, sports, leisure-related activities. Next, a critical aspect of the study is the measurement of the potential mechanisms through which physical activity and exercise may be beneficial. There's evidence from animal studies that at least one pathway for benefits involves an exercise-induced increase in proteins that promote neuron health. These proteins have been found to contribute to learning and memory-related changes in the brain. We will obtain blood samples to measure these proteins that are important for good functioning of neuron health. There's also some evidence from animal studies that these proteins may aid in reducing the abnormal buildup of other proteins associated with Alzheimer's disease, that is, amyloid and tau. Thus, this study will include cerebrospinal fluid and PET measures of amyloid and tau protein. Lastly, we propose that the higher levels of the proteins promoting neuron health and lower levels of the buildup of amyloid and tau will in turn be associated with better neuron health. Thus, we will measure proteins in cerebrospinal fluid that are reflective of the integrity of neuron structure. Ultimately, the proposed pathway from physical activity to better neuron health is expected to be associated with brain structure and memory. So, we will obtain estimates of brain function, brain structure from MRI scans. This image of an MRI scan gives you an example of a slice of the brain as if you were going from front to back. Here you can see structures deep in the brain that are important for learning and memory. Reductions in the size of these brain structures are observed in aging and Alzheimer's disease, which appear to go along with the memory difficulties. Thus, in our theoretical model, we're particularly interested in seeing how physical activity and those measures of proteins and potential mechanisms relate to the size of these brain structures. Finally, we will robustly measure memory through tests involving word lists, stories, and word pairs. Overall, your continued participation will be invaluable in examining how physical activity might reduce the risk of Alzheimer's disease, and eventually, whether over years, those who engage in more physical activity do indeed continue to have less amyloid and tau, larger brain structures, and better memory, making them less likely to develop Alzheimer's disease over time. So we hope together to establish how physical activity may promote healthy aging so that we can age as successfully as Ida Keeling as well. In 2011, at 95 years old, Keeling set the world record in her age group for running 60 meters at 29.86 seconds. In 2016, just before her 101st birthday, she set the world record for the number of push-ups in her age bracket and broke her own world record in the 100-meter dash. She just turned 105 this August and is still running. Thank you for your participation.
Hello, my name is Dr. Joyce Balsbury, or Joy. The title of my presentation is the Overview of the New Health Disparities and Equity Core in the Washington University School of Medicine, Knight Alzheimer's Disease Research Center. My plan is to provide an overview of the new purpose of this new research core that we have in the Knight ADRC. I'll describe health disparities, health equity, and social determinants of health. We'll also determine some opportunities that we have to address health disparities in the St. Louis area through research and talk a little bit about next steps. So I'd like to start first by saying it is wonderful meeting you virtually. So to start, I'd like to say that I am from St. Louis, and I'd like to start with a quote from a Nelly song that says, "I'm from the Lou and I'm proud." So it is the 20-year anniversary of Country Drammer by Nelly. As many of you all know, it's been all over the news here locally. And just a little bit of background about who I am, because some of you may not know me. So I grew up in the St. Louis area in the Central West End, and I am a proud graduate of St. Louis public schools. And my educational journey did not actually start in the public schools. It started at the Wilson School, which is right by the Danforth campus. From there, I went to Cathedral, which was part of the St. Louis Catholic schools. And from Cathedral, I ended up at Sticks for a couple years. And during that time at Sticks, my parents realized that I needed another transition, a smaller school. So I transferred again. So if you're following this, by the time I was 8 years old, I had attended 4 different elementary schools, finally ending up at St. Francis Xavier Elementary School, which was on the campus of St. Louis University. It was at St. Francis that I was diagnosed with dyslexia. So I live every day with dyslexia, and I believe that it is a superpower. And it gives me the opportunity to think very differently from my friends and colleagues. It also means that sometimes folks think that I'm procrastinating, and it's not procrastination. It's just that I'm always processing on how I want to show the good deeds that we are working on. After my 8th grade promotion or graduation, as they called it during those days, I went on to Health Careers High School, which was over on South Grand, just south of Cardinal Glennon Hospital. And during that time that I was at St. Francis, I actually volunteered at Barnes Hospital and actually worked at the hospital 2 days a week as part of my training experience and qualifications for high school graduation. I completed high school in 3 years, which was a family tradition. My mother also completed high school in 3 years, and so did my brother's daughter, my niece. And so we wanted to make sure, and even my mother's sister. So following suit and following the women on that side of the family, I also attended a historically Black college. I graduated from Xavier University of Louisiana with a Bachelor's of Science degree in psychology. After Xavier, I returned home and worked as a church secretary at Lane Tabernacle CME Church at 910 North Newstead. It is my family church, the church I grew up in, the church my mother grew up in, the church my grandmother grew up in, and the church where many of our relatives, including me, were married. During my time back in St. Louis, I eventually worked at the St. Louis Health Department, doing active disease tracing, which we hear a lot about due to the COVID-19 pandemic. But at that time, I was doing active disease tracing of babies that were born to HIV positive moms. It was during that experience that I ended up deciding to do my master's degree in psychiatric epidemiology at Washington University School of Medicine. While there, I had a chance to intern at the University of California, San Francisco, and eventually took a position at UCSF, where I worked both domestically in the San Francisco Bay Area, including San Francisco and Oakland and surrounding areas. And then I also worked internationally in Uganda. And a bulk of that work centered around infectious disease, namely HIV and AIDS, as well as hepatitis. During that time, I had the wonderful chance of meeting a ton of scholars, and I was encouraged to go back to school to do my doctorate degree. I completed my doctorate at the University of Toledo, and during that time, I met a boy, and said boy was from Florida. And he talked me into moving to his hometown of Jacksonville, Florida. So he moved back there before I did. And while I completed my master's in health education and epidemiology, I eventually transitioned to the University of Florida, where I did my postdoc in health equity, epidemiology, and biostatistics. While at UF, I had the chance of meeting some colleagues at Mayo Clinic, and eventually applied for a position at Mayo when my postdoc was ending. And I transitioned to Rochester, Minnesota, where I was one of the founders of the Community Engaged Research Program in the Mayo Clinic Center for Translational Science, as well as eventually my last role at Mayo was serving as the founder of the Diversity and Disparities curriculum for the 5 schools within the College of Medicine and Science. And I also had my own research lab there. I was eventually recruited to come back home to work at Washington University School of Medicine with my mentor, Dr. John Morris. So I'm delighted to be back home. So that picture in the right corner is a photo of my husband and me attending his uncle's or our uncle's birthday party. And it just shows the fact that he always has my back and that he always tries to make me laugh. He really did not want to take that photo, but I snapped it anyway. My husband is a professional musician, and I love all things related to music. He even played with Prince at one time. And so that's why I wanted to start the talk with some lyrics, because we love music from hip hop to gospel to country. So, so there's a little bit about me. So let's talk a little bit about health disparities and health equity. So many times the term health disparities and health inequalities are used interchangeably, but we want to keep in mind that it doesn't relate to all differences in health. But many times it relates to those differences in health that are linked directly to things that we may be able to change, and some systematically, and some things that systemically we are unable to change that relate directly to discrimination, access to care, geographic location, and the like. In the United States, we may hear the term health disparities, but if we're talking about the work that happens in other countries, we'll sometimes hear the term health inequalities. And again, it refers to specific differences in health or other things that impact our health that are sometimes influenced by policy. So to put things in a different light, I also like to share with you some information that was provided by the World Health Organization as it relates to social determinants of health. Social determinants of health really relate directly for the haves versus the have-nots. It relates directly to where we're born, where we grew up, where we currently live, where we work or don't work, and even our age. It also links directly to distribution of money and power and resources at not only a local level, but global, national, and even regional. So what about my hometown? So 87% of St. Louisans have a high school diploma or equivalent. Sadly, close to a quarter live in poverty. Over half of us are non-white, and over half identify as female. 14% of current St. Louisans are 65 and older, but we know that number is going to increase because our population is aging and it's also growing. There are a variety of risk factors that are linked directly to Alzheimer's disease and related dementia, or what some refer to as ADRD. Age is a factor, with the risk doubling every 5 years after the age of 65. If there's a family history of ADRD, our educational level, ethno-racial factors, especially those factors linked directly to discrimination based on race or ethnicity, our socioeconomic status, which is linked directly to education and our wealth experience, our lifestyles, the activities that we do for quality of life and wellness, and those that may hinder our quality of life or wellness. Cardiovascular disease like hypertension, endocrine disease like diabetes, our biological sex, with women being at greater risk, and even brain injury. So there are some key ingredients that I have listed here to increase health equity. It is about thinking about what can we do and what can policymakers do, and even the systematic and systemic changes that can happen in order to increase health equity. Those are based on the actual needs that we have as individuals. Because sometimes what I might need as a woman who's not quite 5 feet even, versus my husband who's 6'3", almost 6'4", are drastically different. So it goes back to our access to care. How close are we to care? Do we have insurance? Do we have proper insurance? It even goes back to workforce diversity, because there's some literature that states that there is a greater comfort level of us speaking to healthcare providers that have had shared experiences, either based on where they grew up or their race or their gender. It goes back to innovation and research and ensuring that all populations and communities are represented at the research table, which leads us into community engagement and thinking about how can we, as scientists, engage directly with communities and patients, physicians and other healthcare providers and resource providers to identify the local needs, the regional needs, the national and even state and global needs in order to achieve health equity and reduce disparities. It goes back to thinking about federal, state, and regional and local actions and laws and policies that are put in place to impact quality of life and health and wellness. It even goes back to us as the individual and behaviors that we may need to modify in order to increase health for ourselves and our loved ones. And then even thinking about precision medicine. What can be done for us as individuals in terms of looking at our genetics, looking at the new treatment mechanisms that might be coming out that will impact our health and ensure that we have a better quality of life. So there is a responsibility that I always go back to as it relates to scientists. One of our big responsibilities is understanding and underscoring the historical underpinnings of mistreatments of minorities, especially African Americans, Native American communities, the Latinx community, and women and others who have been historically mistreated for the advancements of science. This goes back to acknowledging the Cold War experiments that were done in the St. Louis area. It goes back to providing a voice of those descendants who are now living in the shadows of that mistreatment. One of the reasons I decided to come back to St. Louis was the dedication that the Knight ADRC has of addressing health disparities and health and increasing health equity in my hometown by giving a voice to people that look like me, people that sound like me, and wanting to make sure that all people are represented in science and that we're doing what we call good and fair and ethical science. Because our local community is aging. And as I watch my loved ones age past 65, and as I get closer to that, I got a little bit of time left, but as I get closer myself, I want to make sure that the voice of those that I care about are heard and that we have the opportunity to engage in science in a way that is appropriate and that advances the care and well-being of all of us and those around us. So the core purpose of the new Health Disparities and Equity Core in the Knight ADRC is listed here. We plan to use community and patient engaged research processes to reduce ADRD health disparities and increase health equity in diverse communities. This is where we'll need your help. I hope that we're able to engage in a deeper dialogue about what this purpose means so that we can gain insight on areas to improve the health of my hometown. And for some of you, your hometown and the places that we live and engage. So we have three main specific aims. Our first aim is to identify and document current ADRD health disparities related to ethno-racial factors and other social determinants of health and the ADRD catchment area using multiple research approaches. So what that is basically saying is that we want to find out what is currently occurring as it relates to improving health and addressing health disparities in the St. Louis area. And we would do that by doing multiple different types of research studies, from surveys to observational studies to focus groups to key informant interviews, from biomarker studies. And I can keep going on and on about the wonderful opportunities that we have to engage in science. We also want to determine the feasibility of a community and patient engagement research model to address health disparities based on those needs that we identify. And then another goal or aim is to partner with the other cores within the Knight ADRC, Washington University School of Medicine, and other areas to supply training opportunities for the next generation of ADRD, health disparities, and health equity researchers and clinical providers. One of these opportunities will start with the creation of what we call the Co-Equal Registry. Co-Equal stands for creating opportunities to increase health equity and equality for persons at risk for Alzheimer's disease related dementias and related dementias. We are in the process now of writing a National Institute of Health grant application to secure funding to start the Co-Equal Registry. So why a registry? Currently, the Knight ADRC is doing amazing work as it relates to the identification, the prevention, and even the treatment of ADRD work and health disparity and increasing health equity around ADRD. We've had a number of wins, including the creation of the African American Advisory Board, the MAP studies, and the like. But there is one key element that we would like to increase that would work to build on that existing infrastructure to increase diversity and inclusion in our studies. Many of our current research studies really focus on white middle class communities. This means that the communities in which I'm from, and I did grow up in the Central West End. However, as an African American, many times I don't see people in our studies that look like me, or that may look like the greater population of that 52% of non-white individuals that reside in the St. Louis area. So the Co-Equal Registry will allow us to expand some of our efforts by looking at what we call comorbid health conditions. So other illnesses or health conditions that may increase risk for ADRD. And it'll also allow us to think about what are the social determinants of health that we want to address as it relates to ADRD, especially around diversity, inclusion, and what I like to call belonging, so that we can increase that bandwidth of the voice around the research table. So I reviewed for you the purpose of our new core. I've talked a little bit about the aims of the new core and some of the methods. I also wanted to, and hope that I clarified a bit our local efforts. And one of the goals that I have as a new member of the Knight ADRD faculty and leads in terms of the Health Disparities and Equity Core is to increase health equity in ADRD in the St. Louis area. So our next steps involve creating new partnerships internally and externally, and this is where your help comes into play. Creating even a process with our stakeholders to build what we call bidirectional engagement. So not only me talking, but me also listening and having the opportunity to hear directly from you. And then the submission of this Co-Equal grant and determining other funding mechanisms that may help us to increase health equity. So I'd like to thank my mentor, Dr. John Morris, for this wonderful opportunity to speak with you briefly. I'd also like to thank the faculty, staff, and our learners, so our students, our fellows, our residents at the Knight ADRC, the St. Louis community. And if you'd like to reach me, my email address is listed here. And I'm also very active on social media, so my Twitter handle and Instagram handles are also listed. Thank you so much. And I hope that you have a wonderful day.
Good morning. Thank you for attending our virtual participants meeting. Although we'd much rather be with you in person, we're still glad that you're here. My name is Marissa Streitz. I'm a licensed clinical social worker at the Knight Alzheimer Disease Research Center. It's my pleasure today to introduce our center's newest initiative to incorporate lifestyle or life course factors into our investigation of Alzheimer's disease and related dementias. My goal for today is to explain our interest in studying these factors, clarify for you what lifestyle or life course factors and other social determinants of health actually are, and also explain how we will collect this information. It's important to explain why the Knight Alzheimer Disease Research Center is even interested in looking at lifestyle or life course factors. First, as researchers are learning more and more about Alzheimer's disease every year as scientific advancements are made, you could probably guess that the more we learn about this complex disease, the more questions we inevitably have. For instance, some studies have suggested that African Americans have a higher prevalence rate of Alzheimer's disease as compared to non-Hispanic whites. This is particularly interesting since we know that race is less defined by genetics and biology and more so by social and cultural experiences. Another important discovery made over the last several years indicates that some people have these pathological brain changes, such as an increase in plaques and tangles, which are normally indicative of Alzheimer's disease, but they're not exhibiting memory loss. So, in other words, these people technically have Alzheimer's disease, but they don't have Alzheimer's disease dementia. Why wouldn't they develop memory loss? There are many theories to this, but only focusing on genetics or biological factors, are researchers getting the full picture? What if life course factors and other social determinants of health play a role? Next, I will describe what lifestyle, life courses, and social determinants of health actually are. For this presentation, I'm going to use all three of these terms interchangeably. The Center for Disease Control and Prevention defines social determinants of health as conditions in the places where people live, learn, work, and play that affect a wide range of health and quality of life risks and outcomes. I've listed a few factors here, but there are many, many more, including gender, physical ability, and access to healthcare, just to name a few. Because there are so many factors to study, our team at the Knight Alzheimer Disease Research Center needed to find a way to condense these down and look at the most important factors in the context of memory or cognition. To do this, we reviewed what information is already published on this topic. We also spoke with experts in the field about what they're studying and what they're looking at. Also, and probably most importantly, we kept in mind participant burden. We know we ask our participants to do a lot of things, so we don't want to add to your plate unless absolutely necessary. With all this information in mind, we developed a survey entitled The Life Courses Influencing Aging and Dementia, or the LIAD survey. So these are the variables that we are interested in examining as part of the LIAD survey. As you can see below, we're looking at socioeconomic status. That includes your income, even your occupation. We're looking at physical activity, discrimination and stress levels, early life adversity, that's your childhood experiences, whether good or bad, social support, and even your neighborhood. So not only are we asking you questions about the green space in your neighborhood, for instance, where's the closest park to you, but we're also asking practical questions like, do you feel safe in your neighborhood? Along with these questions that we're asking, we are also utilizing the Neighborhood Atlas tool, which helps us look at adversity in neighborhoods block by block. So, when will you be able to take this survey? Well, currently we're conducting a pilot or a preliminary study. So before we roll this out to all 800 plus of our participants, we need to work out the kinks a little bit. We want to make sure that the study is measuring what we want. We want to make sure that the survey isn't too long, that the wording isn't offensive, and that our participants are willing to take the survey electronically in their own homes. Traditionally, you come to the Memory and Aging Project into the office, and a clinician is with you interviewing you. But this will be a little different. With COVID, we're learning to adapt. So are participants willing to take surveys in their own homes on their own time? We're not sure. Well, we weren't sure. But pilot testing began in the early part of September, and even though we're only looking at a small amount of participants, 100 participants, the response has been overwhelmingly positive, and a lot of people have been agreeing to do it. The small percentage of participants who did not participate when asked were mostly due to technology issues, maybe not having access to the internet or to an email address. But overwhelmingly, everyone has been agreeing to take the survey. So we're really encouraged by that. So what's next? Eventually, all of our participants will be asked to take the LIAD survey. It will be part of your clinical assessment and administered longitudinally every 3 years in conjunction with your biomarkers, your MRI, your PET scans, lumbar puncture, etc. We will offer it to all participants following your clinical assessment. We'd like to send it to you as a link in your email, and so you can complete this on your own time. But a paper version will also be available if you are not technologically savvy or not comfortable or don't have access to that type of technology. So that was a brief overview of the Life Courses Influencing Aging and Dementia survey. Thank you so much for your participation and for listening today. Please let me know if you have additional questions that weren't answered on the panel. You may email me at my email address below. Thank you again.
Good morning, everyone. Thank you for the invitation, and I'm very grateful for the opportunity this morning to share with you details about a recently funded study to return research results to Knight ADRC participants. So a quick reminder of the concerns, the main concerns regarding returning research results. So firstly, the tests themselves may not be approved for use outside of research settings. That is, they are generally not yet used in clinical practice. And related to this, it means that some of the information we get from research results is uncertain, and we aren't always sure of its meaning. And there is no information that can definitely tell a person whether they will or will not get Alzheimer's disease dementia. And there are also concerns that telling people their research results could lead to psychological harms like increased worry or anxiety. And lastly, that knowing your research results could actually have an impact on how you do on your annual cognitive tests. So for instance, it's possible that if someone knows they're at higher risk, they might worry more about their memory and actually do worse on their annual tests. So in light of these concerns, we're very happy to tell you that we've been funded for a 5-year research study to determine the impact of returning research results that give people information about their risk of developing Alzheimer's disease dementia. It's a 5-year project, and very broadly, the goals of the project are to offer all eligible participants, which I'll speak more about in a moment, the opportunity to learn the results of some of their research tests. We have developed an educational brochure that contains information on the option to receive results, the pros and cons, some of the limitations, as well as questions to ask yourself before you might decide whether you would want to know or not. We will then return to all participants who want their results an estimated 5-year risk of getting AD dementia based on their own research results. And the study itself, the primary aims of the research study are to determine the psychological impact for participants in terms of whether there are increased worry or concerns, and similarly to determine the impact on the scientific integrity of the study. That is, does knowing your research results make you do better or worse on your annual cognitive tests? So I want to tell you a little bit briefly about the types of results that we'll return and how we will return them. So Dr. Hartz has been really leading the development of a risk calculator, and the calculator enables us to produce a 5-year risk, an individual 5-year risk of getting Alzheimer's disease dementia based on multiple research results. And we believe that this is really the most meaningful way to present information, that is combining multiple results to produce a single risk estimate. So the report that we've designed will start by providing what we call your baseline risk. That is the risk of getting AD dementia based on individual characteristics like age, sex, race, family history, and education. And what that enables you to do is give a sense of how you compare in terms of your risk to other people who are similar to you. We will then provide you with an adjusted risk that gives you your 5-year risk of getting AD dementia after we've added 3 research tests: your PET amyloid scan, your MRI brain image, and your ApoE genetic test result. We will also provide you with the individual results of those 3 tests. So here I'm showing you a sample report. This is not a real report, but just an example. But to give you a sense of what you see at the top is information about your risk compared to people who are similar to you. And then immediately below it, you have a comparison of how that risk has changed now that we've added the results of some of your research tests. So in terms of who will be approached, all participants in MAP and ACS who meet the following criteria will be approached about the study. So first of all, you must have agreed to be contacted about additional research studies, be 65 years of age or older, and you must have had within the last 18 months both a research MRI and research PET scan, and that's because we need those results in order to calculate your risk estimate. And participants must be scheduled for an upcoming MAP annual assessment. So based on this criteria, our plan is that we would mail all eligible participants the study brochure and information about 4 weeks before they're due for their annual assessment to give you time to read the information. And then we would call you about 2 weeks later to see if you are interested in learning more. So in terms of what it will involve, so for those who are interested, and it is entirely optional whether or not you do want to receive your results, it would involve 2 additional visits. At the moment, because of COVID-19, we aren't having any in-person visits. So the plan is that we would offer all of this via Zoom on the computer. And the first additional visit would be to go over the materials, answer any questions you have, and sign an informed consent document. And then the second separate visit would be where you would actually receive your research results, and those would be delivered by a healthcare professional. And we will invite, based on your preference, either a study partner, your close family member, or friend can attend the visit with you. And wherever possible, we'll coordinate with your annual assessment to avoid overburdening you or having a lot of extra visits. So this is quite critical because this is a research study and we need to be able to compare a group who receives their results to a group who does not. The study design means that half of participants will be randomly assigned to receive their research results in a year. So after those who do want their results sign their informed consent, half of people will get their results in about 2 weeks, and the other half will get their results in about a year at their next annual assessment. We will also ask everyone who gets their research results to complete surveys 2 and 6 months after getting your results, as well as follow up at your annual assessment. And we will invite study partners or collateral sources who are willing to take part in the study and ask them to complete some brief follow-up surveys about their experiences. So we're working very hard right now to get our protocol and all our materials approved by the IRB. And our goal would be that we can start recruiting participants in early 2021, so January 2021. And here's just a snapshot of the core research team. You'll see Amy Oliver, our clinical research coordinator, who will be doing a lot of the recruiting and informed consent. Lily Levin, our public health research coordinator, will be conducting follow-up interviews. And you can see Dr. Hartz and Dr. Linenbringer, their photos there. They are the 2 clinicians who will be returning research results initially. Dr. Hartz is a psychiatrist and Dr. Linenbringer is a genetic counselor. And lastly, I would like to say a huge thank you to the many people who have contributed to making this possible, the many people at the Knight ADRC who are listed on this slide and more, and in particular, Dr. Morris, who has been a champion of our work from the very beginning and been very supportive. And most importantly, a huge thank you to the Knight ADRC participants who have contributed in a variety of ways through the ethics committee, through our participant advisory committee, giving us feedback on all of our materials. And really this is to extend a huge thank you to you because it is you who inspires our work. Thank you.